So I want to welcome everyone to this Instruct-ERIC special event. It's the first of a series of virtual meetings that we will have where we will take advantage in some sense of this COVID-19 period to be able to present some new ideas, new technologies that are available from Instruct-ERIC, or soon will be available for Instruct-ERIC, but just to let people know what is going on. And today we have two quite good new technologies, one by Nir London and the other by Gideon Schreiber. Before that, Dave Stewart will give a very short introduction as to what Instruct-ERIC is about. But first, let me just give two short slides. One slide which shows the Instruct-ERIC Center here in Israel. We are hosting this first meeting of these webinars. In our center, we have services that we give through the community, through Instruct, which you'll learn a bit more about from Dave, for protein expression, protein purification, macromolecular crystallization, and EM cryo-stem tomography, as well as bioinformatics. The first four are in the Instruct-ERIC Center here at the Weizmann Institute, with the figure of the main building that we're in, Medical Science Biology building, and the fifth part is in Tel Aviv. Currently, because of the COVID-19, all of these services are provided remotely. Today, we will have a 10-minute introduction of Instruct-ERIC from Dave, the director of Instruct-ERIC, followed by two talks. Each will be 25 minutes long with 5 minutes questions. Let's turn the talk over right now to Dave Stewart, who will give an introduction to Instruct-ERIC.
Thanks very much, Joel. Instruct's been going for some time and is established as a pan-European infrastructure for integrated structural biology. And the idea is to cover the main structural biology methods, as Joel said, covering the whole sort of spectrum. And the facilities are provided through centers which span 9 countries at the moment. And in addition to providing access to infrastructure, the key infrastructure, there's also training and internship possibilities. Of course, all of this has been really affected by the COVID-19 pandemic. And where we can, we're really trying to think creatively about how we offer things by remote access, how we do more innovative training methods and things like that. So, and that's something that I think everybody across the world is really trying to get to grips with now. So again, if people have got ideas, get in touch. This webinar series is then partly a response to that. I mean, everything, we've all become aware of how well these video things can work. So we're now trying something a little bit more serious in Instruct, and as Joel said, this is the first. The idea is to have them monthly, and we've got we hope for a trial period of a year, and we see how it develops. And I should thank the people that have really sort of got this started, provided the sort of drive for it, and that includes Sarah Butcher, Nir London, Stephen Cusack, Jan Steyer, Joel, and a number of people. Thanks very much. And also thanks to Stephanie Chapman in the hub for setting up all the technology. Joel has already said that the idea is to showcase some of the technologies, but really to indicate how the technologies can be used to deal with sort of attack some of the big biological questions of the moment, not just COVID-19, but that will occasionally pop up, I'm sure, because it's been on a lot of people's mind over the last months. So I guess that's really all I want to say. So Joel, back over to you.
Dave, thank you very much. It was an excellent introduction to Instruct-ERIC. So this series of webinars are entitled actually Structure Meets Function, which I think is really the direction, of course, that all of structural biology is going, to try to have structural biology address the functional questions. So our first speaker is going to be Nir London from the Weizmann Institute, who will be describing a new technology that he has developed and has been using together with other colleagues in Europe. And I think Nir will describe this in detail. Nir, you have 25 minutes and then 5 minutes for questions, okay? Please.
Thanks, Joel. Let me share my screen. All right. So hi, everyone. Welcome to this webinar series. It's an honor to start this, what I hope will become a great tradition. And then today I want to share with you the story of COVID Moonshot, which is a global initiative that is going on for since February in an attempt to come up with new antivirals against COVID-19 and relied heavily on Instruct infrastructure and Instruct members. So a little bit of background. Our lab is interested in covalent ligand discovery. Covalent compounds have a lot of advantages over traditional reversible binding compounds, such as increased biochemical efficiency. You typically need lower concentrations of covalent ligands to get to the same inhibitory effect. They have longer duration of action as you need to resynthesize the protein to regain its activity. And they can be very selective if they're targeting a specific nucleophile that's only found in the target protein and not in one of its homologs. Several successful drugs such as ibrutinib, afatinib, osimertinib has been rationally designed recently to use this covalent binding, whereas previously, historically, they were only discovered serendipitously, but they're still challenging to discover or design. And this is the focus of our lab, and we developed several technologies to address this question. The first is a covalent virtual screening method called the Covalent. The second is covalent fragment screening approach and empirical fragment screening approach, which I'll focus on today. Other approaches include covalentizer, which allows you to start from a reversible binding compound and derivatize it. Covalent peptide docking, which allows to install electrophiles on peptide binders for your target protein. And recently we also published on the concept of covalent PROTACs. But as I mentioned, today we'll focus on electrophilic fragment screening. So in this approach, we have a library of electrophiles, mild electrophiles, well, either chloroacetamide or acrylamides, which are not very reactive, and we pulse them in pools of 5, and then we incubate our target protein with each of these pools for 24 hours at 4 degrees to be able to work with proteins that are not super stable. And then we read out the binding through mass spectrometry, intact protein mass spectrometry. And this is what a typical result may look like. In blue, you see the spectrum of the protein, the deconvoluted spectrum, not incubated with any compound. So this is the actual mass of the protein. And in green, you can see the spectrum after 24 hours incubations with 5 compounds. And as you can see, this protein was labeled to 100%, since we don't see any residual non-labeled protein, by a compound which has an adduct mass of 258 Daltons. And it happens to be that this is one of the compounds in the pool, and it exactly matches the delta in mass. And so we can both identify which compound label and quantify the percent labeling. And we have done so for a large number of proteins so far. But in this talk, I want to focus on our COVID-19 target, the main protease of the SARS-CoV-2 virus, Mpro. You can see a structure of it here. It's a dimer. Here you can see the virus life cycle. After the virus enters the cells and translates its RNA into 2 large polyproteins, Mpro and another protease, PLpro, cut themselves, autocleave themselves from the polyprotein, and then process the rest of the polyproteins into the separate units that can then go on and continue the virus life cycle. So inhibiting Mpro and inhibiting this processing step will be an antiviral, could serve as an antiviral for COVID-19. And this is an essential protein for the virus. So to target Mpro, back at mid-February, the teams of Martin Welsh and Frank von Delft at the Diamond Light Source, which are part of Instruct, got the main protease sequence. They were able to clone and produce it, and within 1 week, got the high resolution crystal structure for the apoprotein, which is amazing in my mind. In only a week, we're now going from sequence to structure. At this point, they sent us purified protein, and we used our electrophilic fragment screen to screen 1,000 electrophiles against the protein. And we got a very high hit rate. About 150 of the 1,000 compounds showed labeling to the target cysteine, which means it's a very nucleophilic cysteine. We followed up on more than 40 hits, validating them by dose response. And since our library is mirrored by XChem at the diamond light source, they could immediately soak or co-crystallize both our covalent hits as well as other non-covalent fragments. And by March 5, they collected more than 1,500 different crystals. And within a month from the initial cloning, we already deposited 80 complexes of Mpro with various fragments to the PDB, and we made all the data publicly available. And actually, just this week, the official manuscript was accepted for publication, but all the data was available since February and as a preprint paper. So here's how it looks like. These are all the fragments bound in the active site of the protease. You can also see that some fragments bind at other places, such as the dimer interface, which may be interesting to develop allosteric inhibitor for this enzyme that is only active as a dimer. And you can see within the active site, if we focus, we really exhaustively span all of the different sub-pocket of this binding site with this initial set of fragments. And based on these structures, these gives us ideas on how to possibly optimize and go to the next generation, better inhibitors of the enzyme by merging. For instance, here, this is a covalent fragment in dark green, and this is a reversible fragment in yellow, and there's an obvious merging opportunity here. Same goes for these two covalent fragments that target different sub-pockets. But as you can imagine from this figure, there are many, many different such opportunities, and we needed both to prioritize them and to exhaustively come up with suggestion of what to make next. And at this point, Alpha Lee and his company Postera from San Francisco pitched in, and we decided to do a crowdsourcing platform. So basically within a day, they were able to implement this web platform where people can submit their design ideas based on the data that we published. And we proposed to prioritize their ideas, synthesize and test them. We published this to the social media. And I think basically by the fact that at that point, everyone were at home and were looking for ways to contribute, we got an overwhelming response from medicinal chemists and computational chemists that rose to the challenge and submitted thousands of design ideas, really investing a lot of time and effort and proficiency in this. So here you can see a random selection of some of the design, the initial designs that were proposed. I hope you can appreciate how diverse these designs are. We actually got very few duplicates. So most of the design ideas were unique. To sum up, I think this is a little bit outdated, but it gives you a sense of the magnitude. Within a few months, we got more than 7,000 different designs from 350 different people. We were already able to make, and at this point, I should mention that Enamine, a CRO, a chemical synthesis CRO from Ukraine, really helped us out in this project and contributed resources almost at cost. So they're synthesizing the bulk of the suggestions. So they made by now close to 1,000 compounds. These compounds are sent for testing here at Weizmann and at another group at Oxford. And so far we were able to identify hits in the sub-micromolar range. So just to show you the assays we're using. So here at Weizmann, we're using a fluorescence-based assay. This is a substrate peptide for the protease, which has fluorescein and a quencher. Once the protease cleaves the peptide, the fluorescein starts to fluoresce. You can see here how an uninhibited enzyme looks like, and this is how it looks like with an inhibitor. we have an orthogonal assay based on mass spectrometry where Chris Schofield's group at Oxford are following the mass of the full peptide and the cleaved peptide, and they can extract the rate of the reaction from that. And you can see here for a selection of random compounds that the correlation between the two assays are very nice. So I want to go into a couple of promising lead series that came out of this effort. The first is, you can see here, histidine 163 of the protease, and some reversible fragments from the preliminary crystallographic screen had pyridines or oxazoles, some hydrogen bond donors. They were able, acceptors. They were able to form a hydrogen bond with the histidine side chain. Some of them also had a second hydrogen bond to the backbone of glutamate 166. A second set of fragments, here's the histidine, the glutamate, bound a little bit further away, all of them putting an aromatic ring with some extension, a cyano or a fluoro or a chloro, that you can see here sort of open their own sub-pockets. So in tan, you can see the surface of these fragments, and in white, you can see that a new sub-pocket emerges by these compounds. This is exactly the structure information that fragment screens allow us to make. And so one of the crowdsource ideas based on these pyridines, and actually on one of the covalent compounds that put a chlorophenyl in the second subpocket, was to merge them and have this chlorophenyl aminopyridine. And we made this compound, we tested it, it had an IC50 of 23 micromolar, and indeed it recapitulated the proposed binding mode where it makes all the interactions seen by both of the subpockets. And while 23 micromolar is not that potent for such a small ligand, it is very ligand efficient. And so we published all of this data online in real time. Here you can see screenshots from the Postera website. This is the address if you want to go and check out the data or contribute new ideas. So the inhibition data is also published after each essay, and people can also propose follow-ups. And so far, we tested 150 compounds from this aminopyridine series, and of these, about a fifth had an IC50 below 50 micromolar. And I want to show you a couple of examples. So this non-trivial beta-lactam addition goes into this, what's the so-called P4 pocket, and gives us an almost 7-fold improvement over the base fragment. All of the structures, by the way, that I show here are actual crystal structures, not model, which are determined at the diamond light source. This is another example. So instead of extending towards P4, extending here towards sub P1, and you can see this lipophilic extension also gives us about 7-fold improvement in affinity. And the best compound so far, switching the pyridine into a quinazoline, gave us a 30-fold improvement in binding, giving us a 720 nanomolar inhibitor, which is still very small and efficient. And maybe the nicest thing is that we can, these are not mutually exclusive. We can think of combining these two additional side chains and this quinazoline here to give a merged compound that might be more potent. And this is something that we're actively synthesizing and following up on now. As a second example, I want to focus on a covalent proposed series. So many of the covalent fragment hits that came up from the screen were chloroacetamide piperazine. And you can see that some of them goes into the chlorophenyl pocket that I just described, whereas other heterocycle goes into a different direction, and these can obviously be merged. So these are the IC50s for a few of the compounds from the original screen. So because these are irreversible, their IC50s are much better to begin with, but still they can be improved. So this biphenyl analog, which doesn't have this important chlorine here, is still better than the original compound. A second substitution coming off of the piperazine extends into the lipophilic pocket here and give us a 780 nanomolar inhibitor. And again, we can think about combining all of these different side chains, adding a chloro here, this second phenyl, and this substitution. And this is something that's actively being followed up now. Just as a third example, to showcase another technology from our lab, using the covalentizer algorithm that I mentioned briefly, a student from the lab, Daniel Zeidman, took a SARS-CoV-1 inhibitor. So this was reported back when the original SARS pandemic came out, and there was an available crystal structure for this compound. His algorithm suggested that exchanging the furanil group with an acrylamide will be able to access the catalytic cysteine and form an irreversible bond. And so we synthesized this acrylamide compound by intact mass spectrometry. We showed it's able to irreversibly label the protein. And what's nice about this series is that you can generate the compound using one pot reaction called the Ugi reaction, and you can very easily diversify the different side chains. So here you can see an initial screening of combinations of two various side chains, and you can see that we can reach higher inhibition constant than for the original one. The diamond light source were able to determine so far 11 different co-crystal structure with various side chains, validating the original docking design, all showing the covalent bond formed with the target cysteine. And based on these structures and the biochemical assays, we were able to improve from the original compound while binding irreversibly, did not show an appreciable inhibition. Through substitution of this side chain here, we're able to arrive at a 3 micromolar inhibitor, which is irreversible now, so likely would have a longer duration of action. And this is one of the most promising lead series against the enzyme. Our near-term plans are to, as I mentioned, explore some of the immediate merges that I've showed you. Another technology we're incorporating now is to use free energy perturbation calculation. This is done on Folding at Home, which is a crowdsourced computational platform. Cumulatively, it now has more computational power than the top 10 supercomputers in the world. And so John Kandera at Memorial Sloan Kettering is using this platform to prioritize various substitutions. So here for the amino pyridine chlorophenyl series, we want to test thousands of possible modifications to fill up this small sub-pocket. So Folded at Home users are running these calculations on their GPUs. These are prioritized for synthesis, and hopefully in the next couple of weeks, we'll be able to test the first of these designs. And in parallel, we're also collaborating with several groups around the world to do live virus assays with these compounds to show that Mpro inhibition translates to efficacy in a cellular model. Our goal is to reach efficacy in an animal model, so a preclinical candidate within half a year. We're under no illusion that this will cure COVID-19, but we're very much hope, since the protease is very conserved, that this would be an insurance policy against future pandemics, and this would be a pandemic preparedness project. If someone would have followed up on something like this in 2003, maybe we wouldn't have been in the situation we are today. So just to briefly go over the participating group, there's too many people to state by name, but the main groups are MedChemica from the UK, are providing medicinal chemistry consulting pro bono, which is much appreciated. In Oxford, Frank von Delft group and Martin Welsh and Chris Schofield's group do protease assay, protein production, crystal structure determination. I mentioned enamine, which are responsible for a lot of the chemical synthesis as well as some other bioassays. We contracted WuXi for some chemical synthesis, and Sci Life Sciences contributed a few chemists pro bono to work on this. In the Israel Center, we do the covalent screening, chemical synthesis, and the fluorescence protease assay. UCB Pharma from Belgium contributed medicinal chemistry consulting and computation chemistry support. Postera are responsible for the web platform for crowdsourcing and for machine learning prioritization of compounds. And the Kundera group at Memorial Sloan Kettering are doing free energy perturbation calculation, and the rest of the world for crowdsourcing medicinal chemistry designs. So thank you for your attention. I'll be happy to take any questions you may have.
Thank you very much. This was it's really exciting talk, and I think it's a paradigm for a whole new approach. So maybe before the questions in the chat. Could I ask you one question? What you've done is a dream that has been talked about for some time of what's called generally open source drug discovery. And whenever it was started, there was always the terribly difficult question, which I'm sure you can imagine, is how did you, or how are you planning to arrange all the IP considerations? Let's say you guys discover something that works.
Oh, so I did.
You must have thought about it some.
Right. I didn't mention this throughout, but it's a very important point. So one of the things that we sort of, our main point from the get-go was that this would all be publicly available, open source, no IP. Every design, medicinal chemistry design that is uploaded to the platform is known to everyone. The data is shared by everyone. And it's true that traditional drug discovery companies would never license something like that because they can't protect it and can't make a profit. But I think antivirals and maybe antimicrobial or antimicrobial resistance drugs are in a different, should have a different non-traditional drug discovery pipeline, since the commercial incentives are not aligned for a big pharmaceutical to develop them. I think this is something that should be backed by governments or big philanthropic organization, such as the Bill and Melinda Gates Foundation, since this is in the public interest. If someone would have invested $1 billion in SARS-CoV-2 in 2003, you know, it would have saved hundreds of billions of dollars to the world in this pandemic. So this is something that drug companies are not willing to really take upon themselves. And I hope this would be a successful example showcasing that if we can push this to an actual drug, I hope this can be an example that would ignite additional efforts in other areas where the commercial incentives don't align. So hopefully this will inspire others. Yes.
I think what's nice is that this is not something talked about, but it's something that is happening. So it really can be an example to see how it works. But so far, what you've shown, I think, is extraordinary. Okay, we have some questions already now on the chat. Great talk. Do you see possibilities to test different electrophilic warheads as a way to turn the potency of selectivity of your fragment leads?
So our medicinal chemistry, medicinal design team sort of defined which electrophiles will be allowed for an actual drug candidate. So chloroacetamides are very good starting points, but they're considered too reactive. We're hoping to gain recognition, reversible recognition and potency to get to a point where we can switch them out for acrylamides, for instance, which are well represented in actual drugs, to lower the reactivity and still gain, still have the potency. So this is certainly something that's on our agenda.
Can I ask you another question? In the automatic screens that you've been doing, to plan for the design, did you come across of any existing approved drugs in all those computer calculations?
Yeah, so actually we know of several groups who are pursuing this repurposing efforts. So yes, we intentionally stayed away from that field of approved drugs, and our design considerations were to find something that's very easily manufacturable. So some of the approved drugs, even remdesivir, are very difficult to synthesize and will be hard to globally ship. So for instance, for third world countries, I don't imagine they'll get big, you know, logistically it will be hard to distribute these difficult to synthesize drugs. So examples like this UGI one-pot, one-step synthesis reaction, if we can find such a drug that we can make in a single step, that will be very cheap and very easy to distribute.
Can you elaborate on the 5 virus tests?
For live virus testing, we're looking into cellular assays, what's called cytopathic assays, where we infect cells with the virus, and then we try to rescue their viability using the small molecules. Also, similarly, plaque assays, where we look for the virus forms holes in cell culture plaques, and we try to rescue these. Each lab has their own. There's also another lab that does microscopy-based assay looking at the morphology of infected cells. Each virology lab that has the capability to work with this virus, and these are pretty rare, has their own assay that they specialize in, and we let them do what they're best at.
What have you learned about collaborating with researchers from all over the world from this process, and do it quickly, sharing data, logistics, and so on, just in case you have to do it again?
Right. It's a very good question. I think we as a byproduct of this project, we set up sort of a platform or a mindset maybe that would make the next project maybe easier. So sharing the data, getting ideas from people, prioritizing them, weekly Zoom meetings, as horrible as it sounds. I think we will have some insights into organization of these large collaborative projects, and maybe that's something that's actually actionable in addition to the drug products themselves.
I think is another point that's just very, I think, important, is that you from the very beginning put all the data on the web, and it's clear the journals had no objection to this whatsoever because of the times maybe, but I think this is an example of something which may go forward for more normal times, but we'll see.
I want to, in that vein, I want to point out that Twitter had a very important role in this project, since one of the strategic partners, this Postera company, actually joined after a large Twitter thread from Martin Welsh describing their initial crystallographic fragment screen, so in recruiting, and then a second time in the crowdsourcing effort was, a lot of them were solicited by Twitter. And then a third time, when we actually submitted this this initial work for publication, not the entire work, but the initial fragment screen, the editor in the journal said, I actually watched this develop over Twitter, and I was very, very happy to see it arriving in my inbox. So I think this really highlights the role of social media in the scientific endeavor, and it will only grow larger in my opinion.
What Martin Walsh wrote, I didn't realize it was a comment, not just for private. Joel, in parallel, we are working on repurposing of drugs as well as Mpro and Mpro. Okay, there's a comment, knowing your background, no Rosetta in there, but of course you mentioned this, so you can answer. Go ahead.
So there's, I hid it, but there's one slide that's Rosetta relevant. So in the optimization of the UGI compounds, where we have 3 different side chain we can test, we used Rosetta to prioritize. We modeled all the different combinations with Rosetta and use it to prioritize. But having switched over to small molecules, I keep Rosetta for protein modeling. So. Not as much Rosetta, Alexander.
We'll have one last question. So what is the homology of Mpro protein with other viral proteins? Have you attempted to explore the binding of your most potent compounds with other viral or any other host proteins, either in silico or maybe in vitro?
Yeah, so there's very, very high homology. So between Mpro of SARS-CoV-1 and CoV-2, 96% identity, and I think 98% similarity. And there are a lot of evidence that compounds that inhibit one of them inhibit many of them, maybe not to the same extent, but for instance, drugs for feline coronaviruses showed some efficacy against SARS-CoV-2, and some inhibitors of this Mpro also inhibited other seasonal coronaviruses. So I think any drug that would come out of it has a great potential to be a pan-coronavirus drug. And who knows, maybe you'll also cure the sniffles every winter.
That's what I was going to ask, but there was some work that Michael Rossman did some time ago on developing.
That was the flu. That was against the flu, not against coronavirus.
We had one against the common cold too. Okay, we'll talk later.
That's a rhinovirus, I think.
Okay,
different virus.
You're right, of course. Okay, great. Nir, thank you very much, and thank those who asked questions. It was a lively question time and an excellent talk. So we're going to go on to our second talk of this session, from Gideon Schreiber, who will describe an entirely new technology, a FRAP method, Line-FRAP, which he has been developing in his lab. And I turn it over to Gideon. Gideon is also here from the Weizmann Institute. Gideon.
So I will speak, I would say, on the other half of this, because I'm going to speak here about the micro. So in other words, we heard a beautiful talk by Nir London about this large-scale drug discovery. But at the end, these drugs have to work, and the proteins and everything else has to work in the cells. And one of the main issues of working in the cells is really how they diffuse there, how available they are. And we have shown and worked before that we published a few years ago that clearly a lot of molecules, especially small molecules, can get stuck in the cell and don't really diffuse properly there because they may bind to all kinds of other things inside the cell. So we were wondering how can we best measure diffusion inside the cell, which is an ongoing problem. There are many different methods to do this, and each method has its advantages and disadvantages. We came up with a method called Line-FRAP, which is basically FRAP, but I will show you the differences and to analyze them. And I think we got interesting results that we want to share with you. So first of all, when we think about crowding inside the cell, then there are many forms of different crowdings that we can have here. So a molecule may collide, which is basically hard sphere crowding. So in this sense, it will slow down through the collision, but still diffuse well. We have hydrodynamic interactions between crowders. We have attractive interaction, and attractive interactions is basically what's called soft interactions, because this would be already something where the molecule would have some affinity towards other molecules inside the cell. We have barriers inside the cell that could stop free movement within. We have sealing effect, and so on. All of them will eventually result in differences in diffusion. And FRAP, fluorescence recovery after photobleaching, is a well-known method to measure diffusion. And the method is based, as you can see here, the method is based on it that you bleach a certain spot, you have a fluorescence, either protein or small molecule, whatever is fluorescence is fine. You're doing the bleaching here, and once you do the bleaching, you will wait and measure the time of recovery over time, and then you will get a recovery curve. So here you can see kind of models. So once you do the bleaching, then the molecules inside the bleaching spot will get dark, and then you will measure the time it takes for the fluorescence proteins or molecules to come back inside the bleach spot until you get homogeneous again, and that's what you can see here. And you measure it over time, and then you can analyze it. The question of analysis is a major question. How do you actually analyze it? And in 2012, a good method of analyzing for confocal bleach spots was shown. Very simple method, basically, where you have only three parameters here. RE, RN, and tau. So tau is the most straightforward. Tau is a half time of the bleach recovery. So, and then you have RE and RN. RN is the nominal bleach radius. What is the nominal bleach radius? This is a bleach radius that you get from the microscope. So assuming, and I will show it in a moment, assuming that you have a rigid kind of media where you bleach it and it's not recovering, then you will get a certain RN. RE is a dynamic, effective bleach radius, and the effective bleach radius also relates to the diffusion rates of the measured molecules. Okay, I will now show you the examples of this. So this is now a comparison of the Line-FRAP with the conventional FRAP. And I think the biggest difference really is Line-FRAP, you see, we only scan here a line. So in a conventional FRAP, we are actually measuring a whole square. Now, the difference is in a Line-FRAP, we can do the scanning much, much faster. And we can do it in about a millisecond per scan, which means that we get results very rapid. And the problem is that if we speak about half recovery rates, and the half recovery will happen very often for rapid diffusing molecules like proteins, this will happen in about 100 milliseconds. So this is an example here of a recovery. And you can see here the recovery. And you see that in the Line-FRAP, because we have a dot each millisecond, we actually get many dots, and we can get a very nice recovery curve for a protein. Here is in conventional FRAP, and conventional FRAP will be between 20 and 50 hertz. So in other words, in a second, we can have anywhere between 20 and 50 snapshots. And then you can see that in this recovery time, we actually get very few spots here. So very few frames. In other words, what we measure, our curve fit is not really good. And even worse than this, as I said, there's a very important parameter here. And this parameter is the width of the recovery of the bleached area, which is actually here. You can see here, this is a bleached area, and you have a certain width to the bleached area. This is a very important parameter because, as I've shown you here before, they're actually in square. So if the half recovery time is not, then the radii here are in square functions. So if I don't have this radii exact, then I won't be able to measure the exact diffusion times. So you can see here, this is the result of the bleach area when we do Line-FRAP. And this is, and here it's with conventional FRAP. And the reason that with the Line-FRAP it's so much better is that we can measure the 1 millisecond immediately after we finish the bleach. While here, the measurements, the first measurement will be about 20 to 50 milliseconds after the first bleach because the time it takes to scan it. And as a result of it, we don't scan actually close, but we are scanning, and you can see it here. When we are scanning, it's like scanning over here. So basically, we already have a lot of back diffusion going on here, and then we don't get a nice area of bleach. So now we speak about the RN. The RN is related to the microscope. So here we have an aggregated protein, and now we bleach the aggregated protein. And as you can see here, there's no recovery when we use an aggregated protein. And if we don't have any recovery, we actually can see the result only of the laser of the microscope. And what you can see, that the size of bleach is changing depending on the bleach time. You see, this is 10, 20, and 63 milliseconds, and the size of the radii is increasing. As I said, the radii is in the power of 2. So this is a very important parameter, and this relates to the bleach time. And the reason that this is increasing is because although a laser light is very specific, it's kind of not diffracting, still there is a small kind of refractivity of it, and as a result of it, it is increasing with time. So if you want to get better results, we should do a very relatively short time of bleach to get a smaller circle here. But in any case, we can take it into account. And this is a result here. As you can see from here, you can actually get very nice radii of the microscope. Now, the second parameter is the RE parameter. The RE parameter is much more complex because now we have actually proteins. And the proteins are diffusing also during the bleach. So we have, on the one hand, we have the bleach, but the proteins will diffuse in and out also around it. So instead of getting this really nice bleach area that you will get here, which is very precise, we have a more kind of, in this, we can see it here, there are boundaries, and the boundaries are soft, and you can see it also here. So we get much softer boundaries, and this relates to the RE, and then the question is, how do we measure RE? So again, because we measure it already 1 millisecond after the finish of the bleach, we get relatively good bleach radiuses. And you see the huge difference between the bleach radius if we do it for 10 millisecond and 63 millisecond bleach time. You see here that the bleach radius is much smaller here with a 10 milliseconds, so we get both smaller bleach radius in aggregated sample, but here the difference is even bigger. And as I said, this is an important parameter. The second parameter that we also can see, and this relates to it, that's a diffusion half time, the back diffusion basically. So the tau half is very different for the same reaction exactly if we do 10 or 63 milliseconds. So for the 63 millisecond, we get a slower back time. So the diffusion is slower, and the reason is that it has to diffuse through a bigger area or volume. It has to diffuse through a bigger volume than in 10 milliseconds. So we have two differences here. We have a difference in the RE and RN parameters between 10 and 63 milliseconds of bleach time. And we have a difference here that we see also in the tau half. So now what happens if we combine the two? And this is the nice thing about the Line-FRAP, and here really you can see the power of the Line-FRAP. So we did individual measurements, and each of the measurements is actually an average of 30 measurements. So, but we did a totally independent biological experiment for the same diffusion. This is diffusion here of BSA and bovine albumin. And what you can see here is that we get in the rate of about 60 micrometers square second minus 1. In all cases, there is some difference between the 10 and 63 millisecond, but the difference isn't really that huge. It's quite small. So we get consistency. And the reason that we get the consistency here is because the slower diffusion time and the larger R values compensate to one another. So overall, we get nice values here. While if you look at conventional FRAP, we can see that the values are all over the place. So anywhere between 90 micrometers square second minus 1 and 20 micrometer square second 1. So this is a huge difference in the diffusion times that we get here in the diffusion rate constants that we get here for the same PSA molecule, which really shows the power of the Line-FRAP in this kind of measurements. We also can see nicely how well the parameters are, the parameters of the diffusion time and of the doing bleaching in 63 and 10 milliseconds, how nicely it converges because the way that we calculate it and because that we do our measurements. And here we use different viscosity. And of course, as we know, with viscosity, we get slower and slower diffusion. And this is indeed what we get here when we do now the diffusion times versus the viscosity. You see we get slower diffusion at higher viscosity with the pegs. And the rates are the same whether we use 10 millisecond or 63 millisecond of bleach time, despite the very large differences that you can see here in the measured half-life of the diffusion. And again, this is due to the differences in the radii that we have here for the bleach spot. Now, the nice thing about this method, you can do it not only in buffer, you can do it actually also in cells, where it becomes more interesting. So here we use two different proteins, which are labeled. We can do any kind of label, so it's not important. We can do chemical labeling, or we can do fusion to some fluorescence protein. And these are HeLa cells. And what you see here is a recovery curve. And again, you see here very nicely that we get, again, our radii of bleach area here, the RE. The RN will not change here. So we get a very nice RE effective radius here as well in live cells. We can do the same also in E. coli. And so not only in eukaryotic cell, again, our recovery curves are very nice. And also here, one can see that we get very nice results also for the RE. Overall, this will result in it that we can now compare in different media. We can use this method in order to compare our bleach times And we can see here that the bleach, if he now makes a molecule larger, so this is in PBS for a certain protein, and we can see this is now the same protein fused to YFP, so it will, the size increases. This is about 120,000, and this is, so this is about 60,000, it's about 120,000 in size, and you can see diffusion gets slower. If we compare this now in HeLa cells, so we can compare this to this, and we can see in HeLa cell we get about 2.5-fold slower diffusion. You can see this is a bigger protein. This is for E. coli. In E. coli, we get about 15-fold slower diffusion than we get in buffer, and about 5 times slower diffusion than in HeLa cells. The same is true for the other protein where we did the measurements. So overall, we get nice, consistent result over different media and different. So another thing that we can measure here now is what happened in the same bacteria. So now we take bacteria and increase the osmotic strength of the buffer from 85 millimolar to 1000 millimolar. And basically what we get, you see here, slow diffusion. This is of YFP, the diffusion here. So we get diffusion of about 4.5, which goes down to about 2 micrometer per second square for 500 millimolar salt. So the reason is that the cells get kind of smaller, they get squished in, and because they're squished in, we have now more content inside the cell, and the cell is basically more dense, and so the diffusion is slow. It's interesting to see then when we increase it to 1000 millimolar salt, we don't get diffusion at all anymore here, and the reason is that probably the cells and this is in this case, so you see they really get very small. So it seems that the density is so high now that the 1000 millimolar, we don't hardly get any diffusion at all anymore in the cell. So that's kind of a nice biological thing that we can learn using our method. Now, what about small molecules? And we heard a very nice talk from Nir about the small molecules. So we tested also small molecule diffusion. And in small molecule diffusion, basically, we did quite a lot. I will just show you 2 examples, but we really get surprises. So this is a small molecule diffusion inhibitor. That's a GSK3 inhibitor. You can read it here. It's a serine threonine proteinase. It mediates additional phosphatase molecule into the serine threonine amino acid residues. And this inhibitor has been shown in preclinical trials to have all kinds of protective actions. The question is, what happens inside the cell? So the experiments here obviously were done on one hand biochemically inside the test tube, and on the other hand in animal models. But now what happens inside the cell? And surprisingly, this molecule has a nice fluorescence by itself, so we don't have to add anything to it. When we do now the diffusion measurements of this here, we get the diffusion. First of all, you already can see that the diffusion back is very slow. And from this, we can calculate the diffusion rate of about 0.5 micrometer square per second 1. And this is versus if you look at BSA diffusion, 60 micrometer square. So this molecule is hardly moving inside the cell. So its efficacy inside the cell will also be very low. Now, this is something people don't look at, but efficacy inside the cell, it actually means, and we have shown it in another study that we did, that this will result in the molecule to be hardly active inside the cell, because most of the molecule is not diffusing to where it should go. It's not inhibiting what it should inhibit, because it's just kind of sticking around. Probably it's doing soft interactions with many other molecules inside the cell, so it can not really diffuse much inside. Another example for a small molecule is doxorubicin. So doxorubicin is a well-known anti-cancer drug, which is still in use. Again, it has a good fluorescence that you can measure here. So we measure diclete. It's doing chelating of DNA here. When we look now at doxorubicin, then we see again, we can see this is doxorubicin in PBS buffer, and now these are in vitro measurements. And now we add to it BSA at different concentrations. And as I said, BSA is moving at about 60 micrometer square second -1. You can see in PBS buffer that the doxorubicin also, it's a much, much smaller molecule. It's diffusing actually much, much slower. Now we start to add BSA, and we can see that it's attaching to the BSA, and we get with about a 1-to-1 ratio between BSA and doxorubicin, we actually get a diffusion rate which is similar to the diffusion rate of the BSA. In other words, it seems that now it's hitchhiking on the BSA, and it's using the BSA to move much faster than it would do without the BSA, where apparently it's aggregated, which we also can see in a micrograph here. So the BSA is deaggregating it, solubilizing it. Now, if you look at what happens inside the cell, then we can see that inside the cell it has two fractions, one of rapid diffusion and one very slow. So probably the slow diffusion fraction inside the nucleus, its nuclear is when it's chelated, and then it's free. So it has enough molecules apparently that it can hitchhike with, so it will not get stuck inside the nucleus like it would be just in PBS buffer. So if I summarize my talk here, then diffusion measurements in various environments teach us about the ability of molecules to function in that environment. Here we show the advantage of Line-FRAP to retrieve consistent diffusion rates of fast diffusing biomolecules in any environment in physiological relevant concentrations. Protein diffusion is 2.5-fold slower in eukaryotic cells, 15% slower in E. coli comparison to buffer. High osmotic stress slows diffusion significantly until it gets to almost complete freeze. And small molecule drugs diffuse surprisingly slow, not all of them, but many of them, due to non-specific interactions they will make with the cellular environment, which will reduce activity inside cells. And what we're planning to look at is, can we improve actually the diffusion rate of small molecules inside the cell? So not only using Lipinski rules, but really to kind of improve it, the diffusion inside the cell, so that the molecules will be also more active and have less non-specific activity, which they tend to have a lot. So now I just want, to thank the people. So most of the work really that I showed you here was done by by an excellent postdoc in my lab, the Batra Day, and the work was started beforehand by Agnes Zotter and Felix, but most of the work here is by the Batra, and these are other members of the group and funders, and I want to thank you.
Great, Gideon, thank you very much. It was really, really interesting. So maybe before the other questions, could I ask you one? Since this method can work so rapidly, have you thought about the possibility of using synchronized cells and looking at diffusion rates at different stages of cell cycle, even during mitosis and so on?
Yes, and it's not easy. You are right. It's something that we had in mind because it relates already to what we did before, because we saw the strange behavior of small molecules inside the cell. We were thinking, does it relate to the cell cycle? The problem here is really to get the cells in the exact same stage. So there are methods to do it, but we hadn't the time to optimize it. So for this, we really have to bring all the cells into one stage. As I said, there are methods we didn't have time to do it.
Thank you. Question to Gideon from Nir. For the GSK inhibitor, could it be that it's completely bound to GSK, and in fact, you're measuring GSK diffusion rates? That's Nir's question.
Well, no, because the concentration we are using here quite high concentration. So we are using micromole concentration of the small inhibitor, and it's basically inside the solution. So it goes. We put this inhibitor inside the solution. It by itself absorbs itself into the cell. So you will have high concentrations inside. So these kind of kinases don't have this kind of concentrations like the inhibitor. Else. And even if it would be bound to the protein, it should diffuse much, much faster, because as I showed, the diffusion of a protein is about 100-fold faster, the diffusion rate, than of this molecule. So if it would be bound, it actually would diffuse rapid, because diffusion of BSA, for example, is 60 micrometers square, second minus 1. And this one is 0.5. So if you just would think about molecular size of such diffusion, you are in a molecular size of many millions, but of course, this small molecule doesn't have this molecular size, so apparently it's doing soft interactions with many other molecules.
For the GSK3 inhibitor diffusion, did you compare it to the diffusion of GSK3? Any idea where is stick to make it so slow? Any correlation between the binding affinity of a small molecule and diffusion in the cell?
I think that's an excellent question. No, we did not. Maybe we should get us some GSK. These are new results, and in order to compare, to see, I have no doubt that this is not where it gets done. So the problem, and that's something I didn't show you, but the problem with all the small molecule drugs is that the log p value is high. So small molecule drugs have log p value of 2, 3, 4, something like it, which means that they can be soluble also inside membranes, but also in solution. So and it's important because else they will not penetrate the membrane. But it also means that they are very sticky molecules, are very hydrophobic. So all these drugs are actually very hydrophobic, which means that apparently they will bind a lot of membranes as well, because they come in, they will go out, but they always be in kind of an equilibrium with, because they're very hydrophobic, with anything else that is also can bind hydrophobic things. So we don't know where it gets stuck. By just looking, because we have a movie, so we see it under the microscope, it's not in a specific place. It's not on a specific membrane. We should look at it with higher resolution microscopy in order to identify maybe it's bind to some intracellular membrane where the resolution of us is not good enough. So that's something we definitely should do.
Gideon, so as a kind of a control, did you try maybe a few other molecules that are more hydrophilic to see if it'd be a quite different range than a hydrophobic type molecule, small molecule?
So, the doxorubicin is more hydrophilic. It's also still hydrophobic, but it's diffusing much faster. And as I've shown, it's diffusing at about a rate of almost 20. What's interesting, it actually binds BSA. So that's another example of how small molecules apparently aggregate, in this case, aggregate on itself, because we did it in BSA buffer. But then when you start to add the BSA, then it's deaggregated with the BSA, and the BSA starts to be the carrier of this molecule. So what you can learn from it, that it will be deaggregated probably when you have BSA around, and then the diffusion of it gets suddenly much, much faster because the deaggregation process that is doing. So it's not some solid aggregate. These are very weakly bound aggregates. And this is something very common, actually. It's called phase separation. Phase separation is basically a very weak aggregates that separate in phase, but once you have some binding like to the BSA, it suddenly can move much, much faster. So this would show you that we don't have to talk here about some solid binding or so. You can get phase separation and other processes as well here.
So there's another question. It says, has DOX been previously observed to migrate to the nucleus in cells? And is there a mechanism, not diffusion as such, but active transport by which it is being trafficked or moved there?
Oh, sure. This is very well known. So doxorubicin is very well known that it doesn't stay inside the cytoplasm. Actually, when you put doxorubicin in the cytoplasm, you will see that all of it migrates into the nucleus in a relative short time, within minutes. It's moving to the nucleus. I don't know whether it's an active or a passive mechanism, because inside the nucleus, it will start to bind to DNA. It will chelate the DNA. So then it really gets stuck there, as we also can see in the diffusion measurements inside the nucleus. And that's well known that that's what doxorubicin basically is doing. So I don't know whether it's active, because it just could be an equilibrium shift in this case.
I think we will, if it's okay with you, we'll stop here. I really would like to thank Stephanie, the other members of the Oxford Hub, for really organizing this in a way. I must say, I'm surprised it worked so well in terms of the discussion, because sometimes these Zoom meetings don't work so well. So really, you did great. I'd like to thank Nir and Gideon very much for their clear and very exciting presentations, and in parallel to all the participants and those who asked questions. And Dave, I would recommend we would continue these. I think it's a very important part of Instruct, these kind of webinars, especially when we can't get together in person. Dave?
Thanks very much, Joel, and thanks for fantastic talks, both of them, really good. I think our hope is that we'll rotate around Instruct centres every month, and we'll see how it goes. And these were really good because there was a flavour of the sort of technology, but also the sort of potential impact of these things. And I think what you say, Nir, is about the sort of problem of how we develop responses against these possible pandemics is very interesting, because as you observed, that it was really a market failure that has put us in this place. One would have hoped that given SARS and MERS, by now we would have had effective drugs. So I think it's really important that we use the tremendous power of structural biology, but also in this sort of integrative way, linking in, as you say, we need the neutralization, we need animal trials to push it through. And there must be opportunities for linking in better, I think, up and downstream. I think what was particularly good about about your example was the fantastic buy-in from medicinal chemists, and that was it's absolutely astonishing. So it just shows what you can do. But you know, it's still a way to go, and the challenge is to bring something to the clinic, really, from this point. But really inspiring. Thank you very much.
And thank you, Dave, for your comments. I fully agree, and it's interesting that such a terrible situation can kind of change the paradigm of the way some science is being done. So it's really good. Okay, great. So I think we can end the session now, and I thank everyone for participating. And again, be well. Bye-bye.